So here we're imaging a 35 micron film, and the DNA are very concentrated, so you can't make out individual molecules at all. Now, when you compress the chamber with a lens, until the 2 points at the center come into contact, and you create these very thin regions in that contact area. And so in that image, the height between the two cover slips is about 250 nanometers. And you can very clearly make out individual molecules. You can see their conformations, you can see them wiggling around. That's because that's much thinner than the focal plane of the objective. So that's the basics of Click. Now, what we did in collaboration with Dr. Walter Reiser was to combine Click with these substrates containing nanochannels. So this is the glass substrate that we use as the bottom cover slip of our Click device. And so this has these microchannels here and these reservoirs that would normally be used for driving the DNA through the channels using pressure, but we didn't actually use them for that purpose. We're just using the nanochannels here, which run horizontally between those two big microchannels. And these ones in particular have a 27 nanometer cross section. So that's almost half the persistence length of DNA. It turns out to be totally impractical to force DNA through these using pressure or electric fields. And that's why we're trying to do it with Click. So now this is the device that I designed over the first half of the summer. So we've designed and built this device. And what it does, so here we have our imaging chamber in the center, the lens that deforms the chamber over there, which is attached to this tube, which is raised and lowered using a piezo actuator, which has about 250 microns of travel and 0.3 nanometers of resolution. So this gives us a very tunable, precisely tunable chamber geometry. We also have this orange thing here, an XY translation stage. So we can move the sample around very precisely with respect to the pushing lens. So we can move the sample around and push on different parts of the chamber with the lens. Here's some more images of the device. Here's the microscope objective coming up from below. There's a little window at the bottom where our sample chamber fits. There's a close-up there. You can sort of see that we have a custom microfluidic system for injecting the sample between these two cover slips. Okay, so here's some animations of the technique that we used to do this. So initially, in the top image there, we have our DNA above the microchannels. Now, so since the nanochannels and the top confining surface are separated by a height of about 35 microns initially. So that picture in the top right there is showing that 35 micron gap, which is much larger than the focal depth of the microscope objective. And so now you see tons of DNA molecules that are out of focus, and you can't make out individual ones. Now, on the second image, you can, as we start lowering the lens, you can see the DNA flowing out from that central pushing region. And down here, you can see it's a little pretty faint at the bottom, but you can see the DNA actually entering the nanochannels. This one over there, it's not very clear, but I'll show you some other images that will hopefully be clearer. And so what we're doing here is we're able to push the DNA into the channels from above instead of trying to flow them in through the little tiny holes of the channels from the sides. So here's a wide field image of that chamber. So in this case, we're imaging the light. So this chamber is illuminated by a laser because we're doing fluorescence microscopy, in which case we illuminate the sample with a laser. It fluoresces in a different wavelength, and we filter out the laser wavelength, normally to just view the molecules, so the molecules have their own light source. But in this case here, we're imaging just the laser light in order to characterize the geometry of the chamber. And so what happens, this is the deformed chamber. The lens is positioned right here. You can see those microchannels that I was talking to you before. So the nanochannels in which we'll combine the DNA are going horizontally between those two microchannels. So the lens is positioned there, and you see these fringes, Newton's rings, which occur at integer multiples of lambda over twice the index of refraction. So, and that's the height of the Click chamber, which sort of has a profile like that. And so here, the two surfaces are in contact. Now, if we look out here in that region over there, you can see the DNA are sort of coiled up into blobs. That's at a chamber height of about 600 nanometers. And out here, that's outside of the nanochannels. The DNA are still confined to a very thin film of 250 nanometers. You can see them very clearly. Now, over here in the nanochannels, you can see there's 3 pieces of DNA that look very long. They're fully extended in the channels. So we've succeeded at trapping and fully extending them from above using click. So here's a video of that process. Here we're pushing down with the lens. There's that microchannel on the side. And over here you can see that there are some fully extended DNA molecules trapped in the channels there by the confining lens. All right, so in summary, we've succeeded in combining click with nanolithography to produce a much simpler approach to confining DNA into nanochannels. Now, the one sort of drawback of this approach that we've used here is that the DNA, since we're pushing at only one point with the lens, DNA can only be confined in the channels in a very small region. So we only get, like you saw in that video, we only get like 3 or 4 molecules into the channels, since we're only pushing at that single point. Now, what we're planning to do in the future is to get rid of that top glass surface altogether and just dip the pushing lens directly into solution to contact the bottom surface with nanochannels, and that allows us to use any lens geometry that we want. So we're going to try this using a cylindrical lens, in which case we get a line of confinement. And so that should greatly increase the number of DNA molecules that can be pushed into the channels in one go. And if that works out well, we'll try using an optical flat, so we get an entire big 2D surface to just push them right in. Okay, well, thanks to everybody at the Leslie Lab, particularly my supervisor, Sabrina Leslie, to Chris McCall for his help in device automation, data analysis, and data collection, our postdoc, Jason Leaf, who helped us set up all the optics and microscope, and to Blair Gia, who helped assemble the microscopy chambers. Thanks for listening. Yeah?
Back on the Newton ring slide, what were those white crosses?
Oh, yeah, here, I'll go back to that. No, I won't, my computer crashed. So anyway, if you're, sorry about that. So you remember on that picture there were some big white crosses on the side, but those were, it's just, so that glass substrate with the nanochannels was provided to us by another professor. He had put these gold crosses onto that glass surface just as indicators to find out where the channels are. And they reflect the laser light very strongly, and that's why they're so white. Yeah?
Is it difficult to keep the DNA in the nanochannels?
No, once they're in there, as long as your lens is held down, they're pretty much stuck in there. Yeah.
Right, okay, so you analyze them with the lens.
Yeah, exactly. You have to leave the lens down. Yeah.
How long does a dye last for?
Well, it depends on the laser power. So they undergo something called photo bleaching, so the molecules are tagged with a fluorescent dye, and the fluorescent dye sort of dims out over time as you image it with using the illumination laser. So it really depends on the laser power. Sometimes it can last for minutes. Sometimes it will only last seconds, depending on how much power you're using. In this case, it would usually last a couple of minutes.